Welcome back to the second day of Advances in Proteomics and Metabolomics 2024 Symposium. I'm Princess Senate, and I'm thrilled to be your moderator once again. So today we have a distinct pleasure of welcoming our second keynote speaker, Dr. Christoph Borchers from McGill University, who will be presenting a talk on novel strategies for the absolute quantifications of human proteins. So Dr. Borchers is a renowned expert in the field of proteomics, particularly known for his pioneering work in multiple reaction monitoring for protein quantification. His research focuses on improving and applying proteomics and metabolomics technologies for clinical diagnostics and structural proteomics. So please join me in welcoming Dr. Borchers. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. So please ask questions using the Q&A system to the right-hand side of the video player. So without further ado, I will now hand over to Dr. Borchers to begin his presentation.
Thank you, Princess. Can you hear me?
Yes, we can hear you.
Okay, good, perfect. I don't see that my camera is on, and I don't know how to turn it on, so I'm sorry about this. Thank you so much for the invitation, and I'm glad to be here and to talk a little bit about our latest strategy for absolute quantitation of human proteins. But before I start, I wanted to go back in time. Actually, to the year 2006, when I moved from UNC Chapel Hill to Canada, and one of the reasons was that I was able to be involved in the CP Tech initiative sponsored by NCI, and together with Steve Carr and Lee Anderson, we were very intrigued about using targeted proteomics for absolute quantitation. And we worked quite hard on this, and we had some good papers showing that it's very reproducible. And this was really the main issue at that time, that proteomics wasn't reproducible. But this targeted proteome analysis can be quite reproducible. That means CVs below 20% and even better. And then we were quite happy in 2012 when Nature method called this the method of the year. But what is important is the use of isotopic labeled standards. One of these approaches, what we have shown is very reproducible, is multi-reaction monitoring, as Princess already mentioned. Now other approaches like parallel reaction monitoring is another one, but I mainly will talk about so-called MRM, where we're using a triple quadrupole. And the first quadrupole is like really to select a precursor ion. We are quantifying proteins, but we are analyzing peptides, which we make out of the proteins by proteolysis. Then we pre-fractionate this by liquid chromatography. This goes online into the triple quadrupole. The first quadrupole selects the peptide of our interest. We know exactly when it's coming. And then we selecting this, it goes then into the second quadrupole, which is a collision cell. It's getting fragmented, and then it's getting transferred to the third quadrupole. And at the end of the third quadrupole, only the fragment ion we are selecting is coming out to the detector, and we're getting an ion chromatogram. This is nothing new. It's well known in the pharmaceutical industry, but not for quantifying peptides or proteins, more for quantifying drugs. And what is important is that we're really using internal standards for each peptide we want to quantify. These internal standards are the peptides that have the same sequence, but one of the amino acid residue is isotopically labeled. In most of the cases, we're using lysine or arginine residues, since we're using trypsin as our enzyme. And this gives us several advantages. First, you never can say we have absolute specificity, but we are approaching absolute specificity. Since we are looking at 4 molecular specific criteria, it's MOVC of the precursor ion, the product ion, the retention time, since the standard, internal standard, and the endogenous peptide that need to come out at the same time. And the relative response of natural and cis peptide transitions. What I mean is that the ratio of Y3 from net to Y3 cis should be the same as Y6 to Y6, for example. Then we're using this for absolute quantitation. Since we know exactly the amount and the purity of our peptide standard, then we can really address it how much is really there. In that way that we determine, for example, plasma, the femtomole per milliliter of plasma or microgram of milliliter of plasma, and so on. The very important thing is that then we have the internal standard, we're using it to optimize our parameters, that we achieve that great sensitivity. It goes into the attomole range. And it's very reproducible. This is a key point of using really internal standards. The next slide is actually animation, but it's not working right now. But what I want to show you is that we have analyzed 45 protein targets 12 times. And what you see, the histogram in blue is when you not normalize it to anything. Where we have an average CV of 25 to 30%, which is not bad, but certainly not acceptable for FDA. When you add the internal standard for each peptide, then we improving our CV more to 4 to 6%. This is in green, the green bars. And the yellow bars, when we do it in a more sophisticated way, when we're adding the internal standard in that way that we have a 1-to-1 ratio from the endogenous to the standard. Then we achieve an excellent sensitivity, reproducibilities around the 2 to 4%. So this slide shows the overview about the MIM method development pipeline. It's a targeted approach. So our collaborators or our clients, they're coming with a list of proteins they would like to have quantified. The first step is really to choose the right peptide, which is most suitable for our M approach. When we have done this, then we're synthesizing both the NAT and the cis peptide, purifying this and characterizing the peptide. And then we're using the peptide for optimizing the transition, and we also determine the retention time and all this. And then next is then we see if we can detect the endogenous peptide and are there any interferences. Then next one is a method development or model validation, where we determine the curve matrix, the method variability and matrix effects, stability, endogenous reproducibility, and so on. There's nothing what is magic. It's a lot of work, and it's important that you do this. Do not cut corners. It's quite important to have a robust assay. Last but not least, we determine the reference ranges, and sometimes we turn these assays into a kit that's accessible for the broader community. Here on the next slide, the validation of this asset, which is according to the CPTEC guidelines. A bunch of proteomics people, including me, we came all together at NIH and worked out these 5 experiments, what we think is important to really to have in the end a very robust assay. It has been published several times, and you can look through it. But again, it's not something what is magic. It is just hard work. So here's, for example, the first experiment we need to do the response curve, and in this case we have the NAT, which is in red, and the blue, which is a variable, and based on the ratios, then we can determine the curve, and which should look like something like this, and then we can really from the curve, we know the lower limit of quantitation. This case is 10 femtomole, and the upper limit of quantitation, 20 femtomole. So this is very typical that the linear range is it's a couple of hundred or thousand-fold. It's nothing unusual. But it is important that we determine the curve, and also what is important that we determine exactly the lower limit of concentration, which is within 20% of CV or less. Then Another experiment is experiment 2. The experiment 1 and 2 are quite important. This is addressing the repeatability. And here we determine the intra and interday variability assessment at 3 concentration and at low, medium, and high. And the total variability is based on this formula then. So here's an example, like we have a couple of conditions shown over here, and then we determine the CVs at low, and it's expected that's a little bit more difficult to have like good CVs, but most of them are still below or around the 20% CV, and the medium then goes down, and even further goes down at the high. So then it looks very good if we can manage to perform experiment 1 and 2. We have already a very good assay. We can do the other experiment 3, 4, and 5 to make it to a complete CPTech validated assay. And we have done this, and then we can analyze samples, and we can do it actually in an automated way, which really helps to process hundreds of samples. The workflow is very similar. What we have, what you can see here with plasma. First, you denaturate, reduce, alkylate, and quench, and digest, and then you're adding, when you have the peptides, you're adding the internal standards. It's a quick cleanup, it's a solid phase extraction, and then from there you're going into your UPLC MMMS system. So I want to show you a few assays we have developed over the years. First, this shows a quantitative assay for plasma proteins. In total, we can do this with 270 proteins at the same time. Many of them are proteins which are involved in diseases like cardiovascular or cancer. Other proteins are certain protein classes which are of interest. The apolipoproteins, we have 16 in here. When you see this, then you can immediately, it makes immediately clear that we can determine the concentration over, in this case, over 6 orders of magnitudes. So it means like the protein that's really high abundance proteins like serum albumin, we can detect at the same time at 6 orders of magnitudes lower in concentration, like this protein. This is absolutely doable. So here, this is also an animation, but I don't know if it works. No, it's not. It's not working. I'm sorry. So this is like the list of all the 270 proteins which we have developed in one assay, in one shot. And this means one time is around like an hour, and 94 of these 270 proteins are approved biomarkers. But in some cases, the sensitivity is not good enough. And then the question is, what can we do? We can do offline 2D LC MMMS. In this case, we're using as well UPLC or HPLC, but not at low pH, at high pH. We fractionate this. In some cases, we can actually pool fractions. And then from there, it goes into our low pH LC-MMS system. And this slide shows that we can really extend our protein quantitation range. Now, not only at 6 orders of magnitude, so means like milligrams per mil. Now we can go, everything what is in green, we only can detect after the first fractionation at low pH, at high pH, and now we can go into actually the sub-picograms, into the picograms per mil, higher picograms per mil, into the sub-nanograms per mil. This, of course, requires a lot of work, but you don't need to measure all of these fractions. If it's like around 20 or even 40 fractions, you can do this so-called hard cutting. That means like you are interested in this particular protein, which you cannot detect directly by one-dimensional, but by two-dimensional. You just take the first fraction of the high pH, and then from there it goes straight into the low pH MM assay. So the next slide shows like what we have to do is not just being more sensitive or going deeper into the proteome, covering a greater range of concentration, but also we need to go faster, because where I see proteomics, where it goes into a high high throughput analysis. And so the runtime is quite important, and this new system, this new LC system, and this faster triple quadrupoles like 6495C or 6495D from Agilent. There are others, of course. We can reduce the runtime from 60 minutes, so it means like 24 samples in a day, down by a factor of 4 to 100 samples a day. And this really is the way to do. Like, then really we see like that proteomics will be involved in analyzing hundreds of thousands of samples. So one example where we used the multiplex proteome analysis, and here actually we did both. We analyzed the proteome and the metabolome from plasma from COVID patients before when they got admitted to the ICU. And here the question was like, since at the first COVID wave, the doctor had to make the decision who is getting a bed in the ICU, because, as you know, it was totally filled up, and he had to make a triage decision. And we thought like maybe a quick plasma analysis, both on the proteome and the metabolome, could distinguish between patients that have a very good survival chance or not. And we did this. We got plasma samples from patients at day 0 when they just got admitted to the ICU, and the second day after, and a week after. And then we analyzed we determined the quantity of 140 proteins in plasma and 160 metabolites in plasma. It's all targeted. We knew exactly what we are quantifying. But then we saw that our statistical programs that we have and software packages that do not really can handle this kind of data anymore. And so we use machine learning. Our student used supported vector, and then she came up like with a handful of proteins and metabolites with high predictive value. And then you see like here, when you just look at the proteomics model, that has a p-value of like 10^11. You clearly see the distinguish between survival and death, and metabolomics the same, but it's getting even better if you combine it, as you can see over here. And this shows the area under the curve, the proteomics and metabolomics, and combine it is like really 0.97. And you have like an accuracy even at day 0 when they just getting admitted to the ICU. So this was quite striking, but I think this is where in this direction we are going. Now I wanted to show another example where we used MIM-based protein quantitation, and in this case it was for characterizing or quantifying an antibody which is supposed to be a drug for a certain kind of cancer, and they had a problem with quantifying these antibodies by ELISA. As you know, antibodies and proteins are getting more and more popular for as therapeutics, and mainly how they quantifying these antibodies is by ELISA, just to show like how ELISA works. It's like you have a capture antibody which is immobilized on the surface. Then you're adding your target. It's getting enriched at the antibody, and then you have your detection antibody, which has an enzyme like which is covalently bound at the enzyme at the antibody, and then you perform your reaction, and in the end you get a sigmoid curve to determine the concentration of your protein of interest. The problem is here, if you have structural changes, you might lose the detection of your protein of interest. And this can happen a lot, and it does happen quite a lot when you're using IgG2 antibodies. And here is the structure of IgG2A, and then you have 2AB. You see that's disulfide bridges. They got scrambled, and then they're changing it, and this can lead, and the same as IgG2B, this can lead like to conformational changes, and in the end it might have an effect on the ELISA detection. And this is exactly what happened at this antibody of interest, and it looked like more or less like this, that the antibody goes down, but they only had like an antibody of a detection antibody like for the IgG2A, but not for the others. And then they were quite surprised. Then what they saw is like that their drug is not accumulating according to ELISA. This is what expected at this kind of curve that you see over here. But actually what I saw, like based on ELISA, it's flattening out and it's getting less and less. So then they came to us and asked us, like, can we maybe get an MS-based assay to quantify the amount of our antibody drug in the patient in plasma? And we said, like, yes, I think we definitely can do this. So we selected first surrogated peptides, then we assessed the detectability in plasma, optimized the digestion protocol, determined the linear range, and then in the end we quantified this antibody drug in patient samples and compared this to ELISA. This is the selection of the peptide. We have 2 peptides from the heavy chain and 2 peptides from the light chain. So we synthesized these peptides, the isotopic labeled one and the net one. We need to have both, and after the synthesis, we purifying this and characterizing this rigorously, like with amino acid analysis for determining the total amount and capitalized zone electrophoresis for the purity. Then we optimized, and using these peptides, we optimized the precursor MMC selection, and it finally gave us a very sensitive assay. And now this is a time preparation, always the same: denaturate, reduce, alkylate, digest, and quench, and then it goes from there into our LC MS system. In this case, it's not MIM, it's PRM. We all it's like here, MIM is typically PRM, where you have the full MS spectrum, not selected precursor ions. But after this, you always can you can select an ion chromatogram of certain precursors you would like to quantify. So this is how the peptides look like. The endogenous peptide over here, peptide 1, 2, 3, and 4, and down there the cis peptide, and you see that the retention time is always the same. This is exactly what it should be. Also, the accuracy, the mass accuracy of the fragment ion is quite good in around of 1 ppm, as you see. And this is important. Now, also what is important is that you see the ranking of the transitions from high intensity to low intensity is the same from the endogenous to the cis. So we're quite happy about this. All looks good. And then we optimize the digestion efficiency. We use different denaturants like urea or detergents or TFA, and then we spiked it in and see what kind of time course we can get. And these are the results. In this case, we see that very soon, after around 2 hours, it's flattening out. That means we can use just a digestion time for 2 hours, and then we are totally fine and reach like almost 100%. The next is we determined the linear range, very much the same, but I've shown over there or before. And you see the range is quite good. It goes from 200 nanograms per mil and down to 500,000 nanograms per mil. So it's a huge dynamic range. And what is good, like that means in the end that we can detect clearance of the antibody to 99.9%. So the next is shown is the reproducibility, and you see like all the peptides are quite reproducible. The assays for these peptides are quite reproducible or are below 20%. Then we compare, this is also very important, when you have several peptides, they do not need to give you always the same results, but they need to correlate. And this is what is shown over here. So we have peptide 1 versus peptide 2 with an R square of 1, and all of the other ones are R squares of 0.99 and better. So we are very happy about this. Then the next is to determine the quality of the antibody in human plasma, and then compare this to the ELISA. As I mentioned before, they had this problem with ELISA, but they expected it that it goes up. It should be accumulating over time, but the ELISA did not really show this. But our MS assay really shows exactly what is expected, and this was quite important. Like, and all this, what I showed before, all these determine the reproducibility and linear range and so on. Like, these are very important parameters and characteristics for the FDA. And this is a real-world example where almost this drug failed because of an assay, of an ELISA assay, which could not assess the conformational changes of this antibody over time. Okay, there's always like the conclusion here is it provides this LCP-MS assay provides sufficient sensitivity and detection and quantitation, expected accumulation, and the accurate quantitation necessary to ensure safety and efficacy. So this is important for clinical trials and drug development. Though the next, what I want to say is, in case when we have not enough sensitivity, still, like even if we do 2D, but we also can use antibodies, combining this with our MIM assay. For these antibodies are then there for enriching the proteins of our interest, or the peptides of our interest. And we use this combination and then combining this with more DMS, so immunoaffinity enrichment with more DMS. Here we were able to quantify AKT1, AKT2, and breast and colorectal cancer. We published it a couple of years ago. We also applied the same procedure, in this case immuno-MIM, with precise quantitation of PTEN, another cancer-related protein. And then we also want to use this concept for quantifying PD-L1, which is a protein quite important in immunotherapy. Like, they're using this protein, the PD-L1, as a marker if you are eligible for immunotherapy, yes or no. So all, and this is pretty much tested in every person who has lung cancer. So this means, like, in Canada, over 24,000 every year. And it's important we cannot afford to give everyone PD-L1 therapy because it's quite expensive. We talk about $100,000 per year and per cohort. So then, and if there is no PD-L1, why do you need to give a PD-1 immunotherapy? That's the question want to ask. So we need to have good assays for PD-L1, and there are immunohistochemistry assays, but they're all different. As you see, these are like the pictures, the immunostaining using 4 different kits. So kit A, B, C, and D, and you see that the staining is different. This is like staining from tissues, adjacent tissues, just like 20 micrometers apart from each other. And you see there is, this always gives you a different picture. The immunohistochemistry is not standardized. That's a problem. It's not truly quantitative. There's no clear cutoffs, and the utility is moderate. And then we thought, like, actually, the chief pathologist at Jewish General Hospital, Dr. Ernst Spatz, came to us and said, like, can we do something better? Like, we all know we need to test PD-L1, but the immunohistochemistry assay is based on just what I just said, is not very reliable. Can we get something better? And then we said, like, yeah, I think we can. Another thing is another slide here, which shows, and there's the case study where they have like a disagreement PD-L1 positivity in up to 37% of cases. These are like well-trained pathologists, and still they cannot come up with the same results. Our idea was then we're using mass spectrometry methods, either immuno-MRM or immunomole, which can then be fully standardized. It's highly sensitive, less prone to interference from post-translation modifications. It's truly quantitative. We have the femtomole per micron per total protein and precise. So these are 20%, and of course specific. And we want to not only quantify PD-L1, we want to also quantify proteins which are involved in the PD-L1 pathway. We did this, and we developed this method over here shown, and we can use either formalin fixed paraffin embedded core tissues or fresh frozen tissues. When we have the FFPE, we need to paraffinize it first, and then from there we extracting then the proteins and digest the proteins, adding then the internal standards to each peptide you want to quantify, and then the immobilized antibodies for each peptide we want to quantify. And we can do it in a highly multiplexed way. Then we're pulling out both the endogenous peptide, which you want to quantify, and the internal standard with the antibody. Then we clean it up and elutes this, and these peptides, and from there it goes into our LC-MM-MS system. This worked out quite well, and here are these proteins for which we have developed a multiplex assay. We have 4 peptides for PD-L1, 3 peptides for PD-L2, 1 for NT5E, 3 peptides for PD-1, and 1 for LCK and ZEP17. And if you see, like, the approach is quite sensitive. The lower limit of quantitation goes down to low femtomole or even sub-femtomole. So we're quite happy about this. This is working well. And now we're analyzing some real samples. And what we have seen in our first preliminary results is that we can sort them, these patients, based on the PD-1 and PD-L1 axis proteins. And we have 2 defined groups. One is immune high, survival high, and one is immune low, survival low. And now we are analyzing even more sample, and hopefully we can not only show that this PD-L1 assay based on mass spectrometry not only is technically more suitable than the immunohistochemistry, but also it gives you a higher clinical utility. So now I want to come actually to the topic of to the title of my presentation, where I wanted to talk in the last couple of minutes about the approach which we think will replace how we do this right now. How do we do this right now? We're starting with the discovery approach over here. In this case, we have lung tissue. We analyzing this and doing this label-free and find some proteins which are up or down regulated. Now we want to validate this using a targeted approach. We need to have internal standards. We're synthesizing this, as I said before, both the net and the cis. Then after synthesis, it needs to be purified and needs to be characterized by capillary zone electrophoresis to address the purity and the total amount by amino acid analysis. When we have that, then we can determine the regression curve, and in the end, we can absolutely quantify it when we're adding the internal standard to our sample. It's all good, it works well, but the problem is here, the surprise. For each pair of net and cis, you need to consider it costs around $1,000. But you can do it like it's affordable if it's just like a handful. But if you really want to go into hundreds of proteins which you want to quantify, absolutely quantify, then that is not feasible. This is the direction we are going. On the last HUPO in Dresden, Matthias Menn gave a presentation, and he clearly said what we need next is for each protein, for each human protein, an isotopic labeled protein for which we can use for as a standard on the protein level, not on the peptide level. And then we thought, like, what can we do? And so, like, why not using isotopic labeled mice, the SILEC mice, where all the proteins that have one amino acid residue or more than one isotopic labeled amino acid residues in every protein. Why do you want to do this? You want to use this one for this mouse labeled proteins for quantifying human proteins. And we thought about this. We all know they are very similar in the proteome. The genome is very similar. The proteome is very similar. And we did some bioinformatics exercise. And what we could see is that, to our surprise and our positive surprise, we were very excited about this, that between mouse and human, we have 157,000 tryptic peptides which is being shared. That means, like, we have the same tryptic peptide in the mouse and in the human protein. That means when you have the isotopic labeled mouse, we can use this shared peptide for quantifying the human protein. Then we did some calculations, and we figured out it's around, from the 20,000 proteins what we have, almost 14,000 or 70%, clearly close to 70%, are quantifiable if we're using isotopic labeled mouse. So here is, on the next slide, you see the scheme, and we call it SysQuant for system-wide quantitation. We feed the mice with C13 labeled lysine residue, and this is an essential amino acid residue. That means that you cannot build this up, you need to take it in with your food. And then we take out the organs or whatever we want to analyze, the plasma, and we can use everything from the mouse. It's a good thing. And then when we want to, in this case, want to quantify the proteins in the lung, the human protein in the lung, we take the same matrix, so it means like heavy mouse lung together, put them together at the ratio of 1:1 ideally, and then see how many doublets do we see. The peptide at the lower MOVC corresponds to the human peptide, and at the higher MOVC to the mouse labeled peptide. And this gives us the number of quantifiable peptides or proteins. And then what we do next is when we have seen it, I said like, okay, now we want to know what the absolute amount of the human. We see the doublet. The question is like how much, what's the concentration or what's the total amount of the isotopic level mice. And we do this by quantifying this using non-isotopic labeled peptides we synthesize, which is much cheaper. And you can do it radioactive. So this is the approach we want to apply. We have some preliminary results already, which is shown over here. Here we analyzed liver from the human liver and put it together with the heavy liver from the mouse. And we put it at the same, as early as possible. That means like on the same matrix. And then we digest it like together. And then we separated. In this case, we have like 48 fractions. We want to know how many. The first question: how many proteins we can we quantify? And then from there, we can turn this into a targeted approach, an MRM-based approach. The same we did this with mouse plasma, shown over here. And here are the results. In liver, from the 10,500 proteins which are known in the Matthias Uhn data bank, we can identify 10,000 mouse human proteins, liver proteins, and from that, like 70% we can quantify. Plasma, similar, not as good, but at least like 30% we can, 1,400 proteins we can quantify, absolutely quantify using this approach. So, and then we looked a little bit deeper into the liver proteins we can quantify. Out of the 1,500 known in the metabolic pathway, we see around 1,000. And this is quite important. I want to cite here Gary Petty, who said like, was a metabolomics expert, and said it's not good enough like to quantify the metabolites because these metabolites are involved in different pathways. But now, looking at the proteins, we can combine this with the metabolite expression level, and in the end, to better understand which of these pathways, or in general, the biology which is behind the different expression of the metabolites. So now we're going from there, from the untargeted one, from the discovery one to the targeted one. And the good thing is the untargeted one, like it took us like 2 days. We have 48 fractions. We wanted to see as much as possible. But now we are just interested in certain kind of proteins, and we do it directly without any fractionation. That's the beauty of MRM. And here we choose like 14 proteins which are involved in the metabolic pathway. They're involved in several different pathways of this entire pathway system. And we were able to turn this into an MRM-based quantitative assay. And then we compared the results of the protein quantitation, what is known in the Bernard-Küster database, but here you use only the intensity, and you see it correlates quite well with an R-square of 0.88, and the slope is all close to 1. That means that we're getting very much the same results out of it. So, very, very excited about this. So these are the MRMs, how it looks like. There's a light in human, and underneath is the heavy in mouse. And here, like the third one, like I don't know if you see my cursor, where you have like this, which demonstrates how important it is to have internal standards, because two peaks are really close by, but the peak with the retention time, which is identical to the cis peptide or the mouse peptide, that's the right peptide of our interest. It really shows like internal standards are very important to improve the specificity. So I'm almost at the end. We not only we used like one system, we had like another system. We analyzed the liver and the liver from human and mouse after mix it together on the timsTOF, and as well on the thermal explorers 480. And you can see here like the results are identical, like or very identical, very similar. That means that it's independent what kind of machine you are using. You get the same results, or not only machine, the operator, different operators and different labs. And this is important, that you have developed a method which can be used by different instrumentations. And even like with even the sample preparation was a little bit different. But the good thing is, since you are adding the mouse isotopic labeled protein early on, that means it accounts for variabilities. So I think just like one summary of this cis-quant approach, we think it makes expensive cis peptides obsolete, and we can multiplex quantitation of thousands of proteins, several peptides per protein, and which gets increased confidence. Coming back to the liver, I'm sorry, I need to drink a sip of water. Coming back to the liver, we had like we are able to detect to quantify 7,000 proteins, but we have over 40,000 doublets. That means around like on average five peptides per protein, which really increase the confidence, and it accounts for digestion, efficiency, and matrix effects, and enables both target and non-target absolute quantitation. So I would like to acknowledge here my team, this is the Siegel Cancer Proteomics Center, and our collaborator at Jewish General Hospital, our collaborators from industry, Evocep and Agilent and Molecular U, and last but not least, our funders, shown over here. In particular, I would like to thank Genome Canada and Genome Quebec. And I'm happy to take any questions.
Thank you, Dr. Borchers, for your excellent presentation. We will now start the Q&A segment of this session. So once again, if you would like to ask questions, now is the time to pop them into the chat box on your right. And I'll do my best to facilitate and address as many as possible. So for your first question, do you have any plans on expanding the strategy you mentioned for quantifying post-translational modifications in proteins?
Yes, absolutely. Like first of all, we started with plasma and lung using the cis-quant approach. We want to go to more organs, and then of course, like we want to extend this as well. Like when we see some PTMs for which we can provide an absolute quantitation as well.
Fantastic. And we do have a question from our viewer here asking for your advice. So when collecting biological samples for proteomic analysis, what would you recommend, heparin or EDTA plasma? So in your opinion, what do you think would provide better sensitivity and specificity in your development assays?
I think we did a long time ago a study, and we decided to go to EDTA.
Fantastic. And for your next question, how can MRM be used to improve the sensitivity and specificity of absolute quantification, especially for low abundance proteins?
Yeah, there are like these machines are getting more sensitive and also getting faster. Faster means you can detect more data points at the same period of time. More data points in the end gives you a better peak shape. And this better peak shapes in the end means like you're more sensitive. You can quantify at a lower amount. So there is a huge push from the vendors to be more sensitive, be more faster. So it means they're collecting more data points. And from our side, we are always like eager to develop approaches which improves the sensitivity. I mentioned one to combine immunoaffinity enrichment and MRM is one thing, or 2D-LC is another thing. But hopefully that the vendors are getting so good with their machines that we don't need it anymore.
Thank you, Christoph. I am aware of the time we have left, so I do want to quickly just talk about the cis-quant kit for anyone interested. Is it currently available? And if so, for which organ or biofluid?
It's not available yet, but we plan to make it available next year. And we start with liver and plasma.
Yep, that's fascinating. So while it's not quite ready for prime time yet, so the
Yeah,
potential applications are definitely immense, and we can't wait to see what it does.
yeah.
For your next question, just as a follow up, how many peptides can be quantified within the single LCMRM MS run using the
Yeah.
SysQuant kit?
So with the latest system, we can quantify 500 transitions simultaneously. So see, like we have like for each peptide three transitions, and we're doing the cis and the nut. They're coming out at the same time. So it means like around like 100 peptides or close to 100 peptides we can detect simultaneously at any time. When we have like and the peak of the peptides is a couple of seconds. So we think we can extend the MM assays to thousands of proteins within an hour.
Thank you, Christoph. Before we wrap up, I just have one final question. So, how can this technology be applied to clinical research, such as biomarker discovery and patient stratification? So, what do you think are the challenges and opportunities in translating this technology into clinical practice?
So the opportunities are certainly, since as I mentioned before, it's very reproducible. That means like even small changes, and biology is like they don't. There's not like a factor of 100 changes. Like in most of the cases, like a certain percentage. When your analytical system is that reproducible, then small changes can be contributed to biological changes, and this can be detected now with these kind of systems.
Thank you so much, Christoph, for your insightful answers. Your work is truly groundbreaking, and we're excited to see how proteomics continues to revolutionize the field of biomedical research. So, any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. Thank you once again for joining us, and we hope you found this Q&A session informative. Please stay tuned for more exciting updates in the field of proteomics, and we will see you all again in our next talk.
Thank you so much.